Hello everyone, welcome back to my channel. And today in biology, we are going to discuss the topic enzymes. And enzymes are very important in the human body. We actually work on the basis of the metabolic reactions because those keep us alive, and those metabolic reactions happen just because of enzymes. And in this chapter we are going to discuss two major learning objectives. First one is the mode of action. So we are going to learn about the mode of actions that enzyme can follow, and there is one method which is more acceptable. We are going to learn this in detail, why it is more acceptable. Then in second one, we are going to learn about the different factors. These factors can be the temperature one. These factor may include pH. This factor may include the enzyme concentration or the substrate concentration, and these one may include the inhibitors. So we are going to discuss these one in detail, and we also are going to use to learn. We are going to learn that how different enzymes are used in different techniques, and how the rate of reaction or the rate of enzyme activity can be determined. So let's start with the structure of enzyme first, because if you don't know the structure, you cannot go into the detail of how it works. So how do we define enzymes? Enzymes are actually defined as the biological catalyst. Now, what is the meaning of catalyst? Catalyst is, we use this one in chemistry as well. So catalyst is anything, it's a general word as well, that can increase the speed of something or increase the rate of reaction. Here we are talking more specifically, so we will say the increase in rate of reaction. And the very second property is without being used up, mean there is no effect on the concentration of the catalyst. So these are the biological catalysts. Now when we use the word biological, it means they are made up of some biological molecule. They work in some living organism, and they are mainly made up of proteins, and we say that proteins are further made up of amino acids. So this is the general, you can say, definition of enzymes. So a catalyst is actually that alters the rate of reaction without itself undergoing any permanent chemical change. So let's move to the structure. So what are the different parts of enzymes that help in its action? So the very first one is its globular structure. So the we you might know that proteins have different structure. One is globular, the tertiary, and different one. So globular, as the name indicate, it is something just like a globe, just circular type, or it's the bonding is so complex that the structure appears to be globular. It has a specific 3D structure based on its tertiary or quaternary structure. So we have different categories, like and it is based on the bonding, like what type of bond is present, whether hydrogen bonding is there, and that bonding actually leads to different arrangement of, you can say, the structure arrangement in structure. Number three, it has a very large size, but in that very large size it has a very small active site. For example, if we just generally take a shape as this circle, so there would be one point, for example, this intersecting point. It may act as an active site, and this is the only site that is actually, you can say, performing the function for enzyme. That's why its name is active. So though the size is bigger, but the size of active site, or the site where the reaction can occur, is quite small in size. Then that's the bit that binds to the other molecule. Here is the enzyme structure if we look at the tertiary structure or the globular structure. So here is the general structure. These are small amino acids that join in through a peptide bond to make a long chain. So amino acids are involved. Then there is a part called as active site. So there would be active site, and let's say this one is active site, this one. So only here the reaction can occur. While if you talk about these, the reaction cannot occur here. Now, what is active site? How do we chemically define it? This is the site where the bond between the substrate and the enzyme is possible. So there would be bonding between substrate and the enzyme, and that bonding actually leads to a chemical reaction which further convert the substrate into product. So this one, these dotted lines are actually indicating the hydrogen bond which is present in between uh, the amino acid to convert it into a tertiary structure. So if we look at generally without like the arrangements, like structural arrangement, it looks like something like this. Now, if we say this is catalyst, catalysts mostly act on, or sorry, the catalysts act on hydrogen peroxide, and it converts hydrogen peroxide into water and oxygen. And why it is so necessary? Hydrogen peroxide is not at all friendly for a human body or any living body. It can um, it can activate other reactions. It can be, you can say, toxic for our body. That's why there is a need to convert this hydrogen peroxide into something that is non-toxic for our body. For example, hydrogen and water and oxygen. Water is used in different processes. Oxygen is also used in aerobic respiration. So these are the useful products. So catalysts actually convert hydrogen peroxide, which is a substrate, into the products which are water and oxygen. So all the metabolic pathway. Now what is the metabolic pathway? These are all type of chemical reactions that take place in our body. And those reactions can be catabolic, those reactions can be anabolic. Now let's go into the detail later. So metabolic pathways are controlled by enzymes in a biochemical cascade of reactions. So some are intracellular, some are extracellular. Now what is meant by intracellular? Means they are produced and function inside the cell. For example, if we talk about like mitochondria, it's a powerhouse of a cell. But how does it produce power? Like how can it generate power? Just because of chemical reactions, because they generate some ATP in our body. So through aerobic respiration. So those are actually produced inside the cell and they are used inside the cells. While if we talk about extracellular, there's the enzymes that is, you can say, secreted by one organ but are used somewhere else. For example, you are very well familiar with pancreas. The pancreas release a pancreatic juice which contain different enzymes like lipase, amylase, and the trypsin. So these act in do not act in the pancreas. Rather, they travels to the very first part of the small intestine, which is duodenum, and act over there. So these are the extracellular one. So this is the main difference. So when we talk about the catabolic or anabolic, now if we say lipase, amylase, or trypsin, these are the catabolic one. Now, how do we define the catabolic reactions that are actually cutting something or breaking something? Means bigger molecule breaks down into a smaller molecule. On the other side, they are anabolic. For example, in assimilation. In assimilation, the very small nutrients, for example, glucose or amino acid, they join together to make a bigger molecule. Amino acid makes protein, as we have discussed this in enzyme. So this one is anabolic reaction where there is formation of a bigger molecule or a bigger structure. So this is about the structure of the enzyme. Later on we'll discuss how do they actually work, and we'll discuss both methods or both ways, the induced fit one and the lock and key model. See you in the next video. Stay tuned.